Today we are going to discuss about how nerve impulse are generated and transmitted. When we talk about nerve system in terms of uh, human nerve systems, you can see two types of uh, systems: central and peripheral nerve systems as well. And then we want to discuss about these two separately. The central nervous system and its function, so it's a different one. And autonomous nervous system, and it can be classified into two divisions: sympathetic and parasympathetic division anyway. So in here we are going to discuss about how nerve impulses are generated and transmit in all cells including neurons. Right, ions are distributed unequally between the cell interior and exterior. So, meaning inside and outside the cell, we can see difference in ion concentration. So that means ions are distributed unequally, not equally, unequally between the cell interior and exterior. So, in the same manner, in the same consequences, we can use for these nerves as well. Generally, the inside of the cell is negatively charged, and outside, uh, whereas the exterior is positively charged. Generally, the inside of the cell is negatively charged, and the exterior is positively charged. Generally, in nerve system, in nerve cells. Inside the nerve cells, we can see negative charges, and outside the nerve cell, we can see positive charge. These opposite charges are attracted across the plasma membrane, and as a result, it creates a voltage difference across the membrane. So that is referred as membrane potential. So in this term, you can see that how it's created. So in here we will discuss those things. So now inside the nerve cells we can see in positively negative charge particles, and outside the cell we can see positive charge particles. So that means there is a difference. So that is called voltage difference across the membrane, and that is referred to as membrane potential as well. So these voltage difference. So what is a voltage difference? Ion concentration. So the differences of ions, negative and positively charged particles, right? Comparatively high negative particles we can observe inside the cell wall, and comparatively low concentration of ion, positive high concentration of positive charges we can observe outside the cell. So in here we can see the difference. So this difference is called as membranous potential or a voltage difference. So now you know about the voltage difference, and now we're now going to discuss about the resting potential. In nervous is in rest. So rest means when not sending a signal, or that means non-conducting. So when nervous or neuron is a rest, that means it is not conducting any signals. The membrane potential is called as resting potential. So in a non-conducting neuron, the resting potential is typically between minus 60 millivolt, minus 60 millivolt and minus 80 millivolts. the non-conducting neuron. That means in this moment, these neurons are not sending signals. So in that time period, contain it's they can see resting potential. The value may be minus 60 millivolt and in between minus 60 millivolt and 80 minus 80 millivolt. So the resting membrane potential is maintained. So how this resting potential is maintained? You know, distribution of ion concentration inside of and outside the neuron. So that is what we should discuss. Before that, we will discuss these things. So, distribution of ion concentration inside and outside the neuron. Then, in a non-conducting neuron, the concentration of potassium ion, it's a positive ion, is higher inside the cell, while concentration of sodium plus ion is higher outside. Now, you can see in this picture. In this picture, you can see high amount of potassium ion inside the neuron and lower amount and sodium concentration is higher inside. Inside the neuron, we can see high amount of sodium ion. And outside the neuron, we can see high amount of potassium ion. So in addition, there are chloride and other large ions, proteins inside the cell. So that's why it's become, I think, right now we discuss about these things. So potassium concentration is higher inside the neuron and sodium ion concentration is higher outside the neuron, and chloride and concentration is and chloride and other large ion concentration is higher inside the cell. So as a result, a neuron has a negative charge inside the cell and a positive charge outside the cell. So that's what we discussed so far. Now potassium ion concentration, yeah, potassium is positive ion, but there are some amount of chloride and other large ion as well. So then the proportionate of positively charged ions is higher inside the neuron rather than outside the neuron. So selective permeability of the plasma membrane, uh, selective permeability of plasma membrane to potassium ion and sodium ions. When we talk about this, there are potassium channels and sodium channels, which are membrane bound proteins that are able to open or close in response to stimuli. What we are talking now, the potassium and sodium ion concentration. There are potassium ion and sodium channels. Potassium channels and sodium channels. That means these channels allow to move in and out. There are some channels, so which are membranes bound proteins that are able to open or close in response to stimuli, according to the stimuli supplied by brain or any other stimulant. So these potassium and sodium ion um, can transfer to these channels uh, according to the stimuli. So it is a membrane bound proteins. So potassium channels allow only potassium ion, like a selective permeability. We discussed before now about lock and key theory. You know, in enzyme reaction, right? There is specific sites that allow to um, fix into these enzyme particles. In the same manner, in potassium ion channels only allowed to transfer potassiums, and whereas the sodium channels allow only sodium to pass. That's site specific. So these channels allow to potassium and sodium to diffuse according to their concentration gradients. Now, however, there are more potassium channels open than sodium channels. As a result, there is a net negative charge inside the cell. So that means um, amount of potassium channels open, there are high number of potassium ions channels rather than sodium channels. Now here there are a high amount of potassium channels rather than sodium channels. Now potassium and sodium also the positive particles, no doubt about that. High amount of potassium channels, that means the high amount of potassium enter into these spaces. You can that mean outside of the cells. But sodium ions could not transfer uh, in that way. So that's what happened. So then what will happen? Sodium potassium pump, that's what we are going to discuss now. Sodium potassium pump, this pump helps to maintain sodium and potassium gradient across the membrane by transporting 3 sodium out of the cell for every 2 potassium that it transported in. So after that we are going to discuss about sodium potassium pump. In sodium potassium pump, this pump helps to maintain sodium potassium gradient across the membrane by transporting 3 sodium out of the cell and every 2 potassium that is transported in. That means when this resting potential, 2 potassium ion enter into the cell and 3 sodium removed from these neurons. Then in here, that potassium ion concentration, potassium number of potassium channels higher rather than sodium ions. That means high amount of sodium ion, potassium ion enter into outside, but the low amount of sodium ions enter into uh, these inside the neuron cells. So in sodium potassium pump, the 3 sodium enter outside the cell, 2 potassium ions only enter inside the cells. So these things happen. Then now we are going to discuss about the action potential. The action potential occurs due to the change in membrane potential. So what is a membrane potential or a voltage difference? What is a membrane potential or voltage difference? So we discussed about these things previously. So actually the resting potential or a voltage difference or what we discussed about these things. Sorry, we are going to discuss membrane potential. Membrane potential means what is a membrane potential? The opposite charges are across the path of membrane as a result of voltage difference of membrane potentials. Then membrane potential means the inside the cell is negatively charged, whereas exterior is positively charged. So that is a membrane potential. So action potential occurs due to the change in membrane potential across the threshold value due to stimulants. Now inside the In membrane potential, we discussed about membrane potential. Again, we recall about that. Recall these things. Membrane potential means inside the cell is negatively charged, whereas outside the cell is positively charged. That is a normal condition. Inside negative, outside positive. So what would happen in action potential due to the change of membrane potential above threshold value due to the stimulant? The action potential has three phases, that is called depolarization, repolarization, and hyperpolarization. Depolarization, repolarization, and hyperpolarization. So what is depolarization? A change in a cell's membrane potential such that inside of the membrane is made less negative relative to outside. Depolarization results due to the sodium plus inflow in response of stimulants. In depolarization, negative inside of the membrane made less negative relative to the outside. So what is the reason for that? The sodium ions inflow in the response of stimulants according to the stimulants. So what is repolarization? Sodium channels close blocking sodium inflow. However, the potassium channels open permitting potassium outflow. So this makes the insides of the cell negative. Again, it's the inside the cells become negative. Depolarization, in depolarization, the negativity is become low than inside than outside. So in a repolarization, again sodium channels close blocking sodium inflow, and then again potassium permanent inflow. And however, the most potassium channels open permanent potassium outflow. So this makes inside of a cell negative. That means the potassium sodium ions are not enter inside the cell. So but the same time the potassium, so high amount of potassium channels open and permit into potassium outflow. That means the number of positive ions inside the neurons becomes low, it's become negatively charged. So what is third one? Hyperpolarization or hyperpolarization. So hyperpolarization means that sodium channels are closed, but again sodium channels are closed. That means sodium inflow is not the, the sodium is not permitted to inside the cell. So sodium channels are closed, but potassium channels are open. As a result, inside of the membranes is more negative. So repolarization stage, sodium inflow is so the sodium inflow stop the inflow, stop sodium inflow inside the cell. So admin sodium channels close and blocking sodium inflow. So but potassium outflow is still still we can see potassium outflow. So in repolarization still. So hyperpolarization stem sodium channels still closed, but potassium channels still open. So the inside of the membrane is more negatives than outside. So what is refractory period? Refractory period is a short time immediately after an action potential in which the neuron cannot respond to another stimulus owing to the inactivation of sodium channels. So this prevents the reverse conduction of impulse in an action. After transmission of the nerve impulse, so the inactivation of sodium channels or inactivation of sodium channels means it prevent reverse conduction of an impulse in an action, in action rather, not action. So axon, what is axon? Is a part of the nerve cell. So in depolarization, a chain cell membrane potential, such as inside the membrane, is made less negative than outside. So depolarization results due to an sodium inflow in response to ions. So in depolarization, but after a nerve impulse is transmitted, again repolarization, hyperpolarization, and after the refraction period, no, it's not allowed to transmit any nerve impulse through the any nervous, any stimulus through the nerve cells. So that's what happened. So in here we can see the generation of action potential, a series of action potential that move along an axon is defined as a nerve impulse. So what is a nerve impulse? So in this we can see resting potential, and this is a graph in between membrane potential and time. With this time, you can't see any nerve impulse transmission. So then depolarization and then action potential. Then action potential. You can see the action potential, and again it's become resting potential. So a series of action potentials that move along an axon is defined as a nerve impulse. So what is a nerve impulse? Not only one, but series of action potentials that move along an axon. So it can define as a nerve impulse. So what is a nerve impulse? It is a series of action potentials. Series of action potentials that move along an axon. So the action potential is generated due to sodium inflow. So what is a sodium inflow? It is called as depolarization. What is a depolarization? The negatively charged negativity inside the cells becomes low. Yeah. So the action potential is generated due to sodium plus inflow. In general consider situation, the high potassium inside the cell, inside the neuron, and the low sodium ions outside. So in this depolarization, what happened inside this depolarization? In depolarization, a change the cell's membrane potential such that inside the membrane is made less negative than relatively to the outside. Depolarization results due to the sodium inflow. We can see high amount of sodium ions inside the neuron. So these sodium ions enter through the sodium channels inside the neuron. So that is called as action potential. So sorry. Yeah, it's action potential generated due to the sodium inflow in one location in the action. So this action potential, this action potential spreads to the neighbor location while the initial location repolarizes. This depolarization, repolarization, depolarization, repolarization, depolarization, repolarization. Right? In this manner, in such manner, this depolarization, repolarization process is repeated through the axon. Repolarization, depolarization, Depolarization, repolarization, depolarization, repolarization, depolarization, repolarization. So these things happening along the axon rather. So these series of reactions are called as nervous impulse. Nervous impulse. So this repolarization process is repeated through an action. So that is called as a nerve impulse. So nerve impulse is occurred due to the stimulants. So in this nerve impulse, so you can see series of action potential. In action potential, you can see depolarization, hyperpolarization, refractors, depolarization, hyperpolarization, depolarization, repolarization, depolarization, repolarization. So these things happen throughout the axon, along the axon. So that is called an nerve impulse. The speed of this conduction depends on so many factors, right? And now we are going to discuss, so what are the factors that affected for the speed of this conduction? The first one is diameter of action. You know diameter, you all know about the diameter. See, the conduction speed is increases with an increase in action diameter. When action diameter is higher, the conduction speed also higher. When the diameter is higher, the conduction, the increase with the increase of action diameter. The second one is presence of myelinated action. In you can see myelin sheath in some actions. In some actions we can't see myelin section, myelinated action, myelin sheath. In myelinated neuron, action potential jump from one node to another. You know, myelinated Ranvier node, you can see this the neuron, you can see these nodes. So in myelinated action, so myelinated neuron rather, the action potential jump from one node to another, that means one node of Ranvier to next, one node of Ranvier to next. This can be transmit nerve impulse. So the rate is higher than normal action. So the rate of the transmission or conduction speed is depend on two factors: the diameter of action and then presence of myelinated action. These two more important when we talk about the speed. So then we should discuss about synapses. Actually, it's a very valuable part, and it's a different one. So this is what we discussed today, and we are going to discuss about the membranous potential. So what is a membranous potential? You know, and resting potential and voltage difference as well. So these things are important. In 2019 18 paper, they asked about how nervous impulse generate and transmitted. So this is the answer. And so this is very important.